So by way of introduction, I am originally from Argentina, hence my very thick accent. I have been in the US for a very long time, but I still have a very thick accent. So if you don't understand what I'm saying, don't despair. You'll get used to the accent. It's called adaptation. But also don't be shy and ask me questions. I studied physics and physical chemistry in Argentina, and then I did my PhD in computational neuroscience and neurobiology at Caltech. And then I spent many years at MIT as a postdoc working with Tommaso Poggio. My PhD was with Christoph Koch. And now I've been a professor at Harvard Medical School, and our lab is interested in the intersection between neuroscience and AI, and how we can build smarter algorithms by capitalizing on trying to understand the details of the neural circuitry in cortex, in human and non-human primates as well. We don't work with non-human primates, we collaborate with other people. We do work with doing invasive recordings in human subjects, and I'll say a few words about that as we go along. So the title of my talk is Deciphering Brain Codes to Build Smarter AI. And the basic motto in our lab is that if you can do it, computers can do it too. And all we have to do is decipher how you can solve a specific task. What are the brain codes by which cognition is implemented? So we spend a lot of time really looking at trying to understand neurons, neuronal circuits, and how they implement a variety of different tasks. We focus mostly on questions that center around vision and visual cognition, in part because we can stand on the shoulders of previous giants, and also because we can have a very precise control about the stimuli, and because primates are visual animals, and there's a lot of very rich knowledge about the world that we can extract just from a quick glimpse of an image. So along my slides, you will see some of these QR codes. If that works, you should be able to scan those, and those will lead you to different websites where you can download some of our papers, some of our codes, some of our data. And again, we would welcome feedback if you do engage with our papers, data, and code. So as I said, my slides are mostly an excuse to talk about some of our approaches and to engage all of you in this conversation. Okay, so most of you are quite familiar with some of the enormous successes in AI over the last decade. As my mentor, Tommy Poggio, often says, intelligence is arguably one of the greatest scientific adventures of all times. If we can solve the problem of intelligence, the problem of general intelligence, in principle, we may be able to solve every other problem in science, because we may be able to build machines that are as smart as we are, or perhaps smarter than we are, that can help us with all the other exciting problems in physics, in astronomy, in biology, and so on. So that's partly why many of us are extremely excited about trying to understand what intelligence is and how we can replicate it in machines. So I'm flashing here a couple of success examples that many of you are probably very familiar with. We have now machines that can beat world champions at Go. We have decent initial approaches to self-driving cars. We have devices like Siri who can understand what I'm saying despite my very thick accent. We have algorithms that can thrive in competitions like ImageNet, where you have to label a million images. And yet, despite all of these successes, I think it's pretty clear to all of us that we have a very, very long way to go from these kind of algorithms to the general purpose AI that's depicted in this movie that many of you have seen, perhaps Ex Machina, where we have agents that in some sense cannot be discriminated from humans. In some sense, agents that can pass a Turing test for human intelligence. So many of you are probably quite familiar with the idea of a Turing test. Alan Turing, one of the greatest minds of the 20th century, was a British mathematician. He was very famous for many things. He's one of the fathers of computer science. He was famous for breaking the code that the Nazis were using to communicate with each other. And he conceived the Turing test, which goes more or less like this. So you have, imagine that you have two rooms, and in one of those rooms there's a human, in the other one there's a machine. And the version that he proposed was a question answering system where you pass a question below the door, and then you get an answer. And you can ask anything you want. And then at the end of the day, if you cannot distinguish which room contains a human, which room contains a machine, we say that the machine passed the Turing test. So this is a pretty high bar to pass in many ways. This was specifically formulated in the context of language. We can formulate a Turing test for vision in a similar vein. We can have an image or a video, and you can ask any question you want about that image or video, and we want the answers to be indistinguishable from how humans would answer those questions. So that's one of our goals. We're very far from that. And I'd like to give you a glimpse of some examples of how we're thinking about these problems. And then I'm going to spend quite a lot of time at the end highlighting a lot of the unknowns, a lot of the big mysteries and main challenges in the field that I hope amazing people like all of you guys will be able to help us solve in the decades to come. Okay, so Oscar Wilde said that the greatest events of the world take place in the brain. So everything you see, everything you can understand in terms of language, your feelings, love, your political affiliations, everything happens inside the brain. There are about 10 to the 11 neurons in the human brain, and then that's our challenge, to be able to discriminate what's happening, to understand what are the neural codes, and to be able to translate some of those ideas from biology and from neuroscience into algorithms. So here are a couple of success examples along this route where enormous progress in AI can be traced back to some of the ideas in neuroscience. Of course, perhaps one of the most basic ones is that we talk about neural networks, and the very idea of neural networks is essentially inspired by the idea of neurons that connect with each other. So from the studies of single neuron biophysics, we have come up with computational operations like normalization, or the idea of having a ReLU unit. ReLU stands for a rectilinear operation where you have a threshold and an input-output function that's linear. This is a major oversimplification of the magic that single neurons compute. But some of these basic ideas about the biophysics of individual neurons have inspired some of these computational operations that are commonplace in many algorithms these days. So I already mentioned the idea of going from neural circuits to neural networks. In the visual field, we know that there are receptive fields. There are particular locations in the visual field that neurons are selective for. Those locations tile the entire visual field. And from that came the idea of convolution, which is quite prominent today in many so-called deep convolutional neural networks that many of you are probably very familiar with. Some approximation to neuroanatomy gave rise to the idea of deep hierarchical networks. So instead of trying to solve problems in one step, in what's called a shallow network, or in a single layer or two-layer network, we have this idea that we can concatenate multiple layers of computation. And this concatenation of multiple layers of computation is what gave rise to a lot of the successes in deep networks over the last decade. And this is loosely inspired by the neuroanatomy of the visual system, the idea that you have a hierarchical progression of signaling pathways throughout the visual system. One of the amazing things about brains is how robust they are to all kinds of different types of transformations, all types of changes in the world, all types of internal changes. So our visual system is quite invariant to changes in illumination, changes in the scale, position, rotation of objects. At a completely different level, our brains are quite robust to major injuries. There are many neurons that probably die in our brains every day, and yet our brains can function quite well, despite all of these internal and external changes and challenges. So from this idea of robustness came several notions in computer science, including regularizers and dropout. For those of you who have never heard about dropout is essentially doing what I just told you in terms of neurons. We basically kill a couple of neurons in the neural network during training, and therefore we increase the level of robustness that the system may have, so it doesn't depend exclusively on any one given group. And then there are a certain critical ingredient to training neural networks is learning. And from the study of learning mechanisms in cognitive science and neuroscience came ideas like backpropagation and reinforcement learning, which are essential to everything we're doing these days in terms of learning in neural networks. So we'd like to continue with this list. We'd like to be able to go deeper into the map between neurons and neural networks. So I'm going to talk about this slide, and then I'm going to make a quick stop and just make sure everybody's following. So the particular problem that I'd like to pose today, and one that we and many other people are interested in, is has to do with visual cognition. An image is worth 1,000,000 words. Given an image, we can ask an infinite number of questions about that image. We can ask, what is there? Where is Obama's food? I can ask you to search for all the mirrors. I can ask you what they are doing. Where are they? What happened before? Who are they? What's going to happen next, and so on. So there's an infinite number of questions that I can ask, and you can answer most of these questions relatively easily. And yet we're extremely far from being able to answer these questions with current computational algorithms. This particular image is meant to be somewhat funny, somewhat humorous. Obama is being playful. He's stepping on the scale. So to understand what's going on here, we have to go way beyond what we can do right now in computer vision. We have systems that can probably take this image and recognize Obama quite well. They can probably recognize that there's several different faces in here and so on. But to really get a deep understanding of what's happening in this image, you need to go way beyond that. You need to understand that this person is standing on a scale. You need to understand that many humans are self-conscious about their weight. You need to understand that Obama is exerting a force on the scale. You need to understand that the person doesn't know that Obama is doing that everybody else understands the joke here. So in order to be able to capture even something pretty basic that you can get in a glimpse, in a fraction of a second, you can understand what's happening in this image. You need to go way beyond the information contained in the pixels, connect that to your prior knowledge about the world, and be able to integrate information in space and time to be able to come up with an interpretation and answer these questions or understand why this image is meant to be humorous. Okay, so I'm going to give you a quick glimpse of how we are attacking some of these questions by getting into some discussion about neuroscience in the brain. This very complex diagram here that many of you have seen represents a coarse mesoscopic understanding of the connectivity in the primate visual cortex. So each of these boxes represents a visual area. So here, for example, in the bottom of this diagram, we have the retina. The RGC stands for retinal ganglion cells. Here, this purple region is the primary visual cortex. This is the part in the back of our brains that's essential to convey information from the lower levels onto the higher levels of visual cortex. And each of these boxes stands for a particular chunk of the brain that's responsible for processing visual information. So we don't know yet the detailed connectivity of every single neuron in visual cortex, but we have a pretty decent understanding of the what I call the mesoscopic connectivity, about how these different areas essentially are connected anatomically to each other. And that's what's summarized in this diagram here. This diagram here follows an approximate hierarchy. So information flows from the retina to the LGN, from the LGN to V1, from V1 to V2, from V2 to an area called V4, and so on. So there's an approximately hierarchical progression. It's not a strict hierarchy for many reasons. There are many horizontal connections. There are many connections that go from one area to another in a somewhat horizontal fashion. There are also massive top-down connections. So I told you that information goes from V1 to V2, but also information flows back from V2 back onto V1. As a matter of fact, purely from an anatomical standpoint, there are more connections going back, that is from V2 to V1, than from V1 to V2. So the forward connections from V1 to V2 are much stronger than the backward connections, but the anatomical backward connections seem to be more numerous than the feed-forward ones. So we have this basic skeleton that guides our thinking about how to build models of visual cortex. So this is one of the basic constraints that we have to respect when building models of visual cognition.
A quick question. So
yes,
the way that you're describing it is that there's more connections going from V2 to V1 instead of kind of the forward propagation. I was wondering if algorithms take that into account. I know that back propagation is like important for learning, but like what you're saying, more connections going backwards, like why haven't we accounted for that?
That's a very good question. And I would say that's at the heart of decades of discussion in vision. So there are a lot of our models have mostly been based on the purely bottom-up feed-forward pathway. And we still have much less insights about what these backward connections do and how they do those things. So I'm going to give you a few examples of how we can incorporate some of these lateral, horizontal, as well as top-down connections. But there's much more to do and a lot that we don't know about this. Why we haven't really incorporated this, a couple of things. One is sometimes we like to build the simplest possible models and see how far we can push them. So one approach that we and many other people have been taking is let's try to build simple models. Let's push as far as we can with simple models and then only incorporate more and more complexity in the anatomy if and when we need it. So that's one of the reasons. Another reason is that the functional aspects of these connections has been much harder to study. So it's been harder to study what these backward connections are doing. So one of the things that as an engineer you may want to do is you take a diagram like this. Imagine I give you a circuit like this and you say, well, what the heck is going on? What does this circuit do? So if you are an electrical engineer, one thing you may want to do is to start unplugging some of these cables and say, what happens if I unplug V1 from V2? If I unplug the forward connections, if I unplug the backward connections, and so on. So in biology, that's very, very hard to do. So only now we're beginning to have some tools that may be able to allow us to unplug cables in such a specific fashion. So that's an excellent question. I will say more words about this in a couple of examples. So keep that and then maybe we can come back to that discussion at the end. But that's a fundamental question. Why? What are the back projections from V2 to V1 doing and how can we study that?
Okay, thank you.
I talked a little bit about neuroanatomy. So the other thing that we know is that there are 5 decades now of neurophysiology, that is listening to the activity of neurons inside the brain to try to decipher the functional implications of what these different neurons are doing in different parts of cortex. So many, many years ago, Stephen Kupfler put electrodes in the retinas. So basically this entails having a very sharp piece of metal that goes inside different parts of the neural circuitry and listening to the electrical activity of neurons in the vicinity of the electrode. So neurons are the basic cells in the brain. They communicate with each other via electrical signals, and we can capture that by placing electrodes inside the brain. So in the retina, we can, the idea of the receptive fields was discovered by Stephen Kufler. Fast forward about a decade, Hubel and Wiesel put electrodes in primary visual cortex, this part in the back of our brains, and discovered that there are neurons that show orientation tuning, meaning that they respond more strongly to a bar of a given orientation compared to other orientations. And this inspired generations of neurophysiologists that have been heroically placing electrodes in different places. So there are places in visual cortex that are responsible for detecting motion, whether something's moving to the right or to the left. There are places that are responsible for responding to complex shapes, including chairs and cars and faces and so on. So we have a lot of information now about listening to the activity of individual neurons and how different neurons respond to different visual features that can also help us constrain computational models of visual processing. So here's my plan. I want to tell you about two projects, one that has to do with understanding a fundamental aspect of vision, which has to do with visual pattern completion. The second one is about active sampling, and that will touch upon the question of top-down projections that was just asked, and how we move our eyes to actively sample an image. And then I would like to spend some time at the end talking about all of our lacuna in our understanding, all of the things that are still quite mysterious in the way we understand the brain, and hence in terms of how we can build algorithms. And then potentially Hilbert questions in the field and future directions. Okay, so let me start with pattern completion. Pattern completion is a hallmark of intelligence. This is something that we do all the time. So this is the idea that we have incomplete information about the world, and we need to invent or make up or infer what's missing. So here are a couple of examples. So if I give you a sequence of letters, you can probably infer what's going to come next, even though I didn't tell you anything further about the task. Similarly, I can give you a sequence of numbers, and this one is a little bit harder, but you can also guess what the rule is, and then come up with how to complete these patterns. So here's a few other examples. I can tell you, even though it was raining heavily, John decided to go out without and you can probably guess what the word is. I don't know if this is going to work. You can do this in the auditory domain as well. So even if you've never heard this beautiful piece by Antonio Vivaldi, you might be able to get some sense of how this particular piece of music will continue. You can do pattern completion from minimal information. And of course, we do this all the time. We do this when we read a story. Just from a collection of words, you can infer a whole. From social interactions, sometimes you form impressions, rightly or wrongly, about people after interacting with them for a second or two. So we are able to do pattern completion and inference, correctly or incorrectly, in many cases correctly, by just getting a handful of information and being able to infer the rest. And I refer to this as pattern completion. And we study this problem in the context of visual cognition. So even if you have never seen this image before, maybe you've never been to this particular place, this is the Piazza San Marco in Venice. This is one of those particularly tragic days in the Piazza San Marco when the piazza is completely flooded. And you can probably infer quite well what these objects are. And in some cases, you may only have access to a handful of pixels. You can just see a handful of pixels in this object here, and yet you can infer that that's a chair. And this is the problem that I want to tell you about today. How is it that our brains can make inferences and complete patterns that are very heavily occluded? So here's a glimpse of how we study these problems and how we do behavioral and neurophysiological experiments in order to build better algorithms for visual inference and pattern completion. So we take an image and then we delete a lot of the pixels. In this case, we used a technique called bubbles. Essentially, this is like looking at the world through a bubble, through a small aperture. So in this case, there are about 4 bubbles. The more bubbles, the more information you have. So here are more example pictures of a face seen through these bubbles. And here's the original image that has 100% visibility. So we present these pictures and we do behavioral experiments to assess how well people can recognize these images as a function of visibility. So in this particular experiment, we had 5 object categories. There were faces and chairs and cars and fruits. And I forgot what the 5th category was. But anyway, there were 5 different categories. We present these pictures with different levels of visibility, and subjects have to do a 5-way categorization task to infer what the object is. This task, if you have 100% visibility, this task is extremely easy. So if you have 100% visibility, subjects are essentially at ceiling, so they can perform this task very well. Because there are 5 categories, chance is 20%. And this is the behavioral performance that we measure and how performance is degraded with the amount of visibility. So what's quite remarkable here is the strong degree of robustness in human vision. Even with about 15% of the pixels, subjects are close to 80% correct. So people can recognize these objects extremely well, even with 15% of the amount of information. So this is showing that there's very robust recognition despite heavy occlusion. In other experiments that I won't have time to get into, we also measure how long it takes to do pattern completion. That is how fast you can recognize fully visible images, how fast you can recognize these partially visible images. And we measure that it costs about 50 milliseconds extra to do pattern completion. That is, it requires additional computations to be able to make inferences from partial information. So that's at the behavioral level. So now we want to try to figure out what's happening inside the brain. So most of the work in neuroscience looking at neuronal activity has been done in monkeys, in cats, in mice, mostly because it's illegal to go to a human person and stick electrodes inside their brains. Except in very rare circumstances where it is justified to put electrodes for clinical reasons. So for many years now, we have been collaborating with neurosurgeons who implant electrodes inside the human brain in order to map, these are patients that have epilepsy, so they implant these electrodes in order to map where the seizures are coming from for potential resection. So if somebody has epilepsy, they go to the doctor and the doctor will prescribe anti-epileptic drugs. These anti-epileptic drugs work in about 30% of the cases. In a lot of cases, these anti-epileptic drugs don't work. And then one of the procedures to try to remove the seizure events is to try to remove the part of the brain, to excise the part of the brain that's responsible for the seizures. Because obviously the brain is a very precious tissue. You don't want to just randomly remove any part of the brain. You need to pinpoint exactly where the seizures are coming from. So the neurosurgeons bring the patients into the hospital. They open up the brain. They put electrodes inside the brain. The patients stay in the hospital for about 1 week. And during this 1 week, we have a unique opportunity to investigate human brain function at very high spatial resolution, very high spatial temporal resolution, and very high signal to noise ratio. So this is very different from many non-invasive techniques to study the brain. People study the brain from the outside using techniques like EEG or MEG or fMRI. These are just ways of defining what's happening in the brain, trying to define what's happening in the brain from the outside. This is different from what we are doing here. So here we're going inside the brain and we're actually directly tapping into either single neuron activity or population level activity in the form of field potential recordings. So here on the right, you see an example of the type of electrodes that we're using. And here on in the center, you see an example magnetic resonance image of the brain of one of our patients and the schematic rendering of one of the electrodes going deep inside the brain to listen to activity inside the brain. So by virtue of this collaboration with neurosurgeons, we have the opportunity to interrogate what happens in the brain during a variety of visual cognitive tasks. For example, in the case of visual pattern completion. So let me give you a glimpse of what happens during visual pattern completion. So I'm showing you here one example of the responses of one electrode in a part of the brain that's called inferior temporal cortex. We know that the inferior temporal cortex is central for visual object recognition because lesions in inferior temporal cortex lead to severe deficits in the ability to perform recognition tasks. So for those of you who are working in Jim DiCarlo's lab, this is also one of the main areas that Jim DiCarlo has been studying for many years. So here I'm showing you what happens in this particular area, but in the human brain instead of the monkey brain. So what you're seeing here on the y-axis is the intracranial field potential in microvolts as a function of time. So in this case, each of these gray traces corresponds to a single trial. This is the fully visible condition. And we see that we see a visually evoked response roughly at about, with a peak at around 160 milliseconds after stimulus onset. And for a lot of reasons that I won't have time to get into right now, we know that these are visually evoked signals. We know that these are visually selective signals. That is that they respond to some pictures, but not to others. And we think that these visually selective signals are responsible for our ability to do visual recognition. So now we go on into presenting these heavily occluded images, these images that you have 10 to 20% visibility. And we see that despite the fact that this is a much more challenging task, we still see visually evoked responses. They're not identical to each other because the images are different from each other, but we still see strong visually evoked responses in all of these cases, presumably a correlate of the ability to do pattern completion. I told you that at the behavioral level, it costs about 50 milliseconds to do pattern completion. And here we see a reflection of that also from the inner machinery of inferior temporal cortex. So these numbers that you're seeing here corresponds to the activity, the time at which the activity happens in all of these different example images. So here the peak happened at 200 milliseconds, here at 218, 242, and so on. So compared to the fully visible images, again, it costs about 50 to 100 milliseconds extra time in terms of neurophysiology now to be able to do pattern completion. I'm going to switch now. So neural selectivity is retained despite strong occlusion, and the neural responses are delayed also at the physiological level, consistent with what I told you at the behavioral level. So I'm going to switch now to talk a little bit about computational models and how we can use these behavioral and neurophysiological constraints to build models. Any questions so far about the behavior or the neurophysiology?
I have a quick question. So, yes. Since the patients that typically present with this, they're pretty rare samples, is there a particular, I guess, demographic, like is it typically children that are the patients in this, or do they range across ages?
Yeah, so that's a very good question. So the patients range over ages. We have worked with children. We have worked with 20-year-olds, 30-year-olds, 40-year-olds. So I think it ranges basically from 2-year-olds all the way to 40-year-olds. Because this is brain surgery, and for a lot of reasons, people rarely do these surgeries in very old people. So typically, there are very, very few cases of this type of surgery in patients that are, let's say, 60 and above. So the mode of the distribution, I would say, is around 20, and it ranges anywhere from 2 to 40. And we have seen a wide range of patients along this spectrum. Does that answer your question?
Yeah. Thank you.
I was wondering how long does it take, like when a subject is shown the image, for them to respond without the occlusion?
Right. So I think you're asking at the behavioral level. So behaviorally, it takes about 400 milliseconds to provide an adequate behavioral response to the image. it takes about 50 milliseconds extra for the occluded images.
Okay, thank you.
So one sort of minor comment that that I think is interesting and relevant is that there's a pretty big gap here between the neurophysiology and the behavior, in the sense that it takes quite some time from the neural signals all the way to behavior. You can force people to respond faster. One of the fastest behavioral signals that you can elicit is by making eye movements. So people have shown that you can do selective eye movement responses in about 200 milliseconds or so. So we think that a lot of the behavioral delay, a lot of the reasons why sometimes it takes 300 or 400 milliseconds to implement the behavioral response has to do with decision making and with orchestrating the motor response itself rather than with vision itself. So schematically, we divide the problem into vision, visual recognition, decision making, and then executing the response. So of those 400 milliseconds, I think a lot of that has to do with the motor response itself and the decision making. But the visual signal happens at around 150 to 200 milliseconds as shown here.
I have a question that sort of reminded me with what you just said. So if using your eyes as a way to respond behaviorally is faster and is more representative of the time frame that it is to look at something, understanding, and then do a behavioral task, why? Because from what I understand, it's not that common. So is there a reason for why using vision as the behavioral data isn't as common as using, like, I don't know, a lever or a button?
Yeah, that's a good question. So historically, in monkey neurophysiology experiments, people have done a lot of tests with eye movements. So historically, it used to be harder to measure eye movements. It's pretty easy to measure a key press. Anyone can code, to detect a key press, whereas detecting eye movements used to be harder. Now it's becoming easier and easier. So my guess is that we will see more and more paradigms in measuring eye movements. The other thing is that for humans, it seems to be a bit more natural and easier to, if I tell you, whenever you see a chair, press the left key. Whenever you see a house, press the right key, rather than telling you, for a chair, move your eyes to the left or move your eyes to the right. But it's absolutely doable, and you're quite right. I think in the future, particularly if we're very interested in these timing constraints, measuring eye movements is only faster.
I have a question.
Yes.
How does this study we see right here deal with, like, the arbitrariness of choosing how to occlude the image? Because I can imagine that, like, it looks like they made a decision to leave, like, a very significant outline of the face. And I assume that if they had, like, done something like shading completely from the top down, it probably would make it much more difficult to pattern recognize than it would if they left, like, an outline like they did.
Yeah, that's a very good question. So first of all, I should clarify that, so here there are bubbles. So the only constraint was that the bubbles had to be overlapping with the object. So we didn't want to have bubbles that show only the gray background. But there was no explicit intention to keep the outline. However, I think you're right. I think by virtue of placing a number of bubbles randomly, there is some part of the outline that's still present in here. So at the behavioral level, we have done many studies with different forms of occlusion. One of the caveats of these physiological studies is that we cannot just study every possible form of occlusion. So one has to make some decisions. And as you said, these decisions are somewhat arbitrary. We studied one form of occlusion. We're not studying every possible form of occlusion. At the behavioral level, we have studied many other forms of occlusion. And if you send me an email, I'll be happy to send you some of the literature on that. But you're right. I'm sort of making the inference here that pattern completion is pattern completion no matter what. But in reality, I think you're right. I think that there may be how exactly you occlude the patterns may actually have an impact in visual recognition. And also another aspect is that which features of the image you occlude also matter. So if you leave, there are some features that are certainly more diagnostic for some images. And this is something that we and many other people have studied. So it's not just, I'm showing percentage visibility, but that's not the only variable that matters. There are many other variables that matter, and you're quite right. Okay, so we want to build a computational model that can do the same thing that humans do. That is, we want to build the model that can do pattern completion. So I told you about behavior, I told you about physiology. Let me also mention an anatomical aspect of cortex, which is that there are massive horizontal connections within cortex. So I told you that information goes up. I told you that information goes down. There are also lots of connections that go horizontally. That is, within a layer, neurons are connected with each other. So we put together the behavioral cost, the 50 milliseconds, that suggests that we need additional computations, the 50 millisecond cost in terms of physiology, which also suggests additional computation, and the neuroanatomical evidence suggesting that there are slow horizontal connections in cortex, to try to build a model that would include these horizontal connections to do pattern completion. So this is what I showed you before. This is the human behavioral data performance as a function of visibility. First of all, let me show you just an off-the-box deep convolutional neural network. Here's one particular neural network called AlexNet. The details don't matter too much. You can plug in your favorite deep convolutional neural network here. The results are approximately similar. So here we take this pre-trained deep convolutional neural network. We use exactly the same images that the subjects were saw, and then we measure the performance of the top layer in this network. The top layer is called FC7. So we see that FC7 can recognize objects well above chance in this task, but their performance is well below human level. So there's still a big gap with respect to human performance in this purely bottom-up model. So our recipe here was to incorporate horizontal connections. So we take, in this case, one layer, layer FC7, and we connect all the units in FC7 in an all-to-all fashion. So we add horizontal recurrent connections everywhere in layer FC7, and we build what's called an attractor network. These are networks that have attractor states in, that have been shown many years ago to be able to do to solve problems of pattern completion. This was introduced by a person called John Hopfield, and these are known in the literature as Hopfield networks. So basically what we're doing is we're taking a deep convolutional neural network, in this case AlexNet, and we're imposing at the top an attractor network, a Hopfield network at the level of FC7. And when we do that and we train these recurrent weights, we can recover human level performance. So this is an example of how we can take existing ideas in computer science, in this case, a deep convolutional neural network that has been extensively used for visual recognition. We can incorporate basic inspiration from behavior, neurophysiology, and neuroanatomy, and we can come up with a better model that can better approximate human level performance in visual pattern completion. Okay, so I'm going to move on to a second example that I want to give you about the intersection between neuroscience, behavior, and algorithms. And this has to do with how we move our eyes and how we do active sampling during visual search tasks. So I already showed you this image before. Let me see if this video works. So this is measurements. This is showing measurements of eye movements during 12 seconds when somebody is just passively moving her eyes while watching this image. So this is a characteristic of visual eye movements. There are these ballistic movements of the eyes from one location to another. These are called saccades. These saccades span several degrees of visual angle, and they happen about 3 times per second, more or less. If you have never thought about eye movements, this may come up as a surprise to you. Most people don't even know, they're not aware that we are moving our eyes. So it turns out that we're constantly moving our eyes. 3 times a second, we're moving our eyes. And we don't realize that, and the world seems pretty stable despite these super fast ballistic eye movements, because the brain has mechanisms to stabilize the image and to compensate so that we don't see massive blur in the image when you're moving your eyes from one location to another. For those of you who are used to, so who know a little bit about photography and are used to taking pictures, you know that if you don't hold your camera steady and you move your camera, there will be a huge amount of blurring. So there's an enormous amount of movement that we have in our cameras, in our eyes, every time we move our eyes, and yet we're completely oblivious to that because the brain has mechanisms to stabilize the image and compensate for that. So we want to study what guides these eye movements and how, when, and why we make these eye movements so that we can instruct and build computational models to make eye movements the way humans do. So one of the basic reasons why we make eye movements is that we don't have full resolution everywhere in our image. And this is also something that may come as a surprise if you've never thought about this, because the world seems to be perfectly high resolution everywhere. Every time you look at some particular location, you don't see locations that are blurred. Everything seems to be perfectly high resolution. This turns out to be an illusion, and it turns out to be completely wrong. There's only a very small fraction of the image, which is called the fovea, which is the place where you're fixating, where we have high resolution. And everything else is extremely blurred. And the easiest way to demonstrate that is by trying to read outside of the fovea. So I don't know in this shared screen how much you can actually see here, but if you can see this big letter R here, I want you to fixate on the R. Do not cheat, do not move your eyes, okay? So just look at the R and try very, very intently not to move your eyes, okay? And now I want you to try to read another word that's several lines below. Of course, it's easy to cheat, it's easy to move your eyes and read that word. There's nothing magic about that. What's extremely hard, as a matter of fact, I would say basically impossible, is to fixate here and read another word that's very far. This is because you only have high resolution, the kind of resolution that you need for reading at the fovea, and everything else has an eccentricity dependence, that is that your resolution drops very, very sharply when you go outside of the fovea. As a matter of fact, this is so pronounced that in the periphery, that is very far from the fovea, all of us are legally blind. So if you have the same amount of vision in the periphery, if you translate that into the fovea, you would be considered to be legally blind. We have very, very poor vision in the periphery. We only see very, very well what we're fixating on. So it's interesting to conjecture that having high resolution requires a lot of real estate in terms of neurons, requires a lot of neurons to be able to process information. That means that it's very expensive for the brain to be able to process information at this high level of resolution. There are strong constraints for the brain in terms of size and also in terms of energetics. The brain is very expensive from an energetic viewpoint. So if we wanted to build a system that has high resolution everywhere, our eyes would have to be enormous, our brains would have to be enormous, and that would introduce a lot of other problems in terms of survival. So the way evolution solved this problem is by having a very small region where we have high resolution, and then allowing us to move our eyes and quickly and actively sample the image. Okay, so we studied this question about eye movements in the context of visual search. So many of you are probably familiar with visual search. You have to search for your iPhone, you need to search for your car keys. Often it's a very annoying visual problem, and it turns out that it can be quite challenging. Many of you probably have seen this kind of Where is Waldo kind of games, where you have to look for Waldo. It's extremely difficult to do that. You need to be moving your eyes all the time until you actually find the object that the target object you're searching for in this image. So there are 4 key properties of visual search that we want to explain. Oh, I forgot to point out, yeah, so here, everybody gets very upset if I don't actually point out to where Waldo is. So Waldo was right here. Anyway, so the 4 key properties that we want to explain are visual selectivity, that is our ability to distinguish the target from distractors. In this case, we're looking for Waldo. We want to distinguish Waldo from all the other objects in this image. In real world visual search, we need to be able to do this in an invariant fashion. That is, when you're looking for your friend in a crowd, or when you're looking for your car keys, we're not looking for a pixel level exact template match. Your friend may be wearing sunglasses. She may be wearing makeup. He may have grown a beard. He may be wearing a hat. We don't know exactly what, we have an idea of what a person looks like, but we need to be able to have invariance in visual search. We need to be able to incorporate different levels of illumination, occlusion, different colors, different kinds of transformations. So that's the invariance problem in visual search. We want to be able to do visual search in an efficient fashion. That is, we want to be able to do rapid visual search. We don't want to do we want to avoid exhaustive exploration. We don't want to go through every pixel in the image to be able to find the target, okay? And the last thing that I want to mention is that we want to be able to have a computational model that can generalize. That is, I can tell you to look for a particular object that you have only seen once, and yet you can look for that object in an image. For example, I can show you a new face that you have never seen before and ask you for that, to look for that face in a crowd. Or I can show you a character in a language that you have never seen before. Maybe you don't speak Chinese or Arabic or Hebrew. I can show you a character in a foreign language and ask you to look for that particular character. So you want to be able to generalize and do visual search with novel objects that you have never seen before. So sometimes in the AI literature, this is called zero-shot learning. That is, from a single instance, you want to be able to generalize. So we conducted a series of visual search experiments that go more or less like this. First, we have people fixate, then we present the target objects. So here the subjects are looking for a cat. And then we present a search image with, in this case, 6 different objects. And subjects are instructed to move their eyes until they find the target. And what you saw here is a short example of how that looks like. So people are start by fixating. I'm going to advance to the next slide, and I wanted to give you a glimpse of how people move their eyes. So we conducted a series of different experiments with object arrays, which is what I showed you, also with natural images, where people are doing visual search in natural images, and also just to torture our subjects with where's Waldo type of experiments, where people are moving their eyes to find Waldo. So let me jump, in the interest of time, let me jump right into the computational model that we built, inspired by some of the visual search behavioral data that we collected, as well as some of the existing neurophysiological data. I heard that some of the people here are working with Bob Desimone. Bob Desimone has done a lot of seminary work looking at the neurophysiological responses in different parts of visual cortex responsible for attentional modulation during visual search. So we incorporate those ideas from neurophysiology and behavior to build the following computational model. So first of all, we have a target image. This is what we are going to look for. In this case, you're looking for a horse. You may be looking for your friend, your car keys, Waldo, etc. So that image goes through a deep convolutional neural network. This is a schematic rendering of ventral visual cortex. You can think of this as AlexNet, VGG16. This is just a jargon for different types of neural networks that have been extremely successful in computer vision. We store the features of what we want to look for in a module that we loosely refer to as prefrontal cortex. There has to be working memory. There has to be some sort of memory of what you want to look for. And we implement that in a module that stores this information that we refer to as prefrontal cortex. Next, we present the search image. The search image goes through the same exact computational ventral visual cortex architecture. That is, we see the image with the same eyes, with the same cortex, with the same machinery. And then to do visual search, we introduce top-down modulation. So this is one example. Someone asked at the beginning about these top-down connections. This is one example of how we implement top-down connections in a visual cognition task. So we modulate the responses in the entire visual image based on the information about the target that we have in memory. Essentially, what we're doing is convolving the entire image to see which part of the images are more similar to the target. After we do that, we come up with an attentional map, which basically indicates the similarity of different parts of the image with the target. We take the maximum of that attentional map. We use a winner-take-all mechanism, and that's going to be our first fixation. That's going to be how the model moves its eyes. So the model moves the eyes to the winner-take-all location of the attentional map. If the target is there, that's great. Visual search ends. If the target is not there, we go back to the attentional map. We inhibit the previous location. We use what's called inhibition of return. So we don't want to go back to the same maximum as before. So we go to the next maximum. And then that way we can generate a sequence of eye movements for the model and compare this sequence of eye movements with human visual search. So here I'm going to show you first behavioral data from humans and then how this model performs in visual search. So what you're seeing on the x-axis is the fixation number. So in this case, there are 6 objects. So I'm showing you fixation 1, 2, 3, 4, 5, 6. If you were to move your eyes randomly, the dashed line here indicates your cumulative performance. So you would have 1 in 6 chance of finding the target object in the first fixation. You will have 100% chance of finding the object in the 6th fixation. So this is the cumulative performance, the function of fixation. This is human level performance. So even in the first fixation, humans are above chance, and in general, humans can search these objects more efficiently than by randomly moving their eyes. So humans are moving their eyes several times a second in an intelligent way, depending on the features of the target to do visual search. Next, we do exactly the same thing with our computational model. So here's a biologically inspired computational model that is not specifically trained with these images. I forgot to mention that we're not doing any kind of training with human data or with these images, and yet the model can successfully capture the essential ingredients of how humans move their eyes in terms of visual search. Okay, so I'm going to stop there in visual search, and I want to move on to the last couple of slides that I wanted to discuss, mostly giving you a sample of a lot of the things that are quite mysterious and that we're still quite far in terms of our understanding in computer vision.
How do you train the models? I don't think I understood.
You didn't understand it because I didn't say it. So it's my fault, not yours. So we actually have minimal training here. And this is because we wanted to do zero-shot decoding. So we didn't want to have the model memorize the particular object. So this part of the model, the ventral visual cortex, is pre-trained on ImageNet. So ImageNet is this very large collection of images. There are 1,000 image categories, about 1,000 pictures per category, a total of about 1 million pictures, and the models are trained to do 1,000 way categorization. These images are unrelated to the ones that we are using in our experiment. So we take the pre-trained ventral visual cortex and we don't train it at all. So this is completely fixed, this part of the architecture. We tried different architectures. In this, the particular results that I showed you were done using an architecture called VGG16. So for the aficionados, this is VGG16 pre-trained on ImageNet. And then
the representation is completely random weights.
They're not random weights. So all of these weights are trained on ImageNet. So all of these weights are trained to do visual recognition of ImageNet images. So this is a very successful
categories.
With 1,000 categories. That's right. But we never trained this model with our images or with the human data. Okay, but it was trained. All of these weights are not random. All of these weights are trained to do 1,000 way categorization in ImageNet. And then we impose this top-down modulation, and then we impose the winner-take-all mechanism and the inhibition of return. So there's essentially no training here. We're not training with human data. We are imposing in a somewhat ad hoc way all of these different arrows. We're imposing this top-down modulation. We're imposing at which level in the hierarchy this top-down modulation happens. we're imposing the winner-take-all mechanism, we're hard-coding this inhibition of return. But we're not training the model with our pictures or with the human behavioral data. And you can
are you inputting like the category from that was outputted by the ImageNet?
No, we're not. There's no input about the category. So the model takes this image. At the end of the day, we have, in this case, 512 numbers. Those numbers correspond to the features at the top level, and those are used in a top-down fashion to modulate here. We're not using the categorization of this image. We are using all the weights. So all of these weights here are essential to transform these pixels into these 512 numbers. But we're not using any of the labels. The network doesn't know that this is a horse. The network can search for Hebrew characters. It can search for Chinese characters. It can search for Waldo. It can search for any object that it can come up with because it doesn't use any categorical information whatsoever.
And you train the search image on ImageNet too?
This search image goes through the exact same network, but again, there is no training. So the model has never seen this search image before. This is the very first time that the model sees both the target as well as the search image. This network here, this ventral visual cortex module, is exactly the same one as this one, and this was pre-trained on ImageNet. All of this was pre-trained on ImageNet, but we're never training with this image. We're never training with this image. We're never training with any of the human behavioral data either.
Okay.
There's a lot more details in this paper here. So feel free to scan this QR code or send me an email, and I'll be very happy to send you lots more details about this project. Okay.
Yeah. So something just kind of occurred to me. I was thinking, well, like when we train a network like this, there's like a finite number of labels, and I know one of the pillars you described when we try to create these like intelligent models is to generalize well. I was thinking, like, isn't that an inherent problem because we have like a finite amount of labels? Like, how do these models or machines generalize to categories they've never seen?
I think that's a very good question. I think that's not a problem here because this model can generalize very well because we're not trying to do visual recognition here. So if you show me an object that the network, for example, unfortunately, I don't think I have the slide here, but this network was never trained with Waldo. The network doesn't know what Waldo is. It's never seen Waldo. Waldo is not one of the images in ImageNet, okay? But here, we're not asking the network to say, this is Waldo. We're asking the network to locate where Waldo is. So this network can generalize very well and deal with the Waldo images. We show Waldo, it goes through this series of steps. We have this set of numbers, and then we can go through this entire procedure and say, the network thinks that Waldo is here. So it can localize Waldo, even if it has absolutely no clue what Waldo is. So you can present any image that you want in here, any search image, any target image, and the network will do its thing and try to locate the object. Now,
yeah.
the problem that,
I guess I was thinking about something kind of separate from this specific thing that you were presenting.
Right, absolutely. So let me take a step back. So a different question that we are very interested in is asking, what is this object? So this is not locating a target object, but asking, it's about object recognition and what the object is. And in that case, you're absolutely right. So if I give the net, let's say that a network was trained to recognize 1,000 categories, all the network can do is put a label to those 1,000 categories, nothing more and nothing less. So if you show a picture of Waldo to VGG16, the output will be one of those 1,000 categories. The network will say, this is a dog, or this is a car, or whatever the closest image is, okay? Now, it turns out that if you ask humans what an object that they've never seen before is, they may still struggle. They may still be able to describe it in some way, but they may not be able to put a word to it either. But I think you're absolutely right, and you're pointing to something that's very important, which is how do we generalize to novel objects and novel shapes? How do we learn novel objects, novel shapes in systems that have a fixed number of categories? So so last week was the annual Computer Vision Conference, CVPR, and one of the hot topics right now is essentially exactly your question, and this is called open set recognition. So everything that people do when they have a fixed number of categories is called a closed set. You have a fixed number of categories. So now people are starting to think about how do we deal with open sets, where you learn how to recognize chairs and tables, and then all of a sudden you're shown a car, and you've never seen a car before. So you don't want to say that's a car, that's a chair. You don't want to say that's a table. You want to say that's something new. And that's something new that doesn't look like car or chairs. And then you want to start asking, well, how can I learn what that new category is? So that's called open set learning. And that's a very hot topic as of last week in computer vision.
Okay, I think that was a great explanation. Thank you.
Okay, so let me flash quickly. I want to end with, I gave you a couple of examples of how we can use neuroscience and behavior to successfully advance computational algorithms. I want to end on a discussion of many of the current challenges and how far we are from really achieving human level visual recognition. This is not ending on a pessimistic note. On the contrary, I think that this is ending on an optimistic note, because that means that there's a lot of exciting questions for all of you guys to work on. So let me flash quickly a couple of these examples. So the first problem I want to highlight is the problem of how we incorporate contextual information in visual recognition. So if you have never seen before one of my talks, and I ask you, what is this object? In the interest of time, I'm not going to stop here and ask you what it is. Trust me, in general, it's pretty hard to recognize these objects. Some of you may guess what this is, but it's not that easy. If I show you the entire image here, so you've got access to full contextual information, then I would contend that it's pretty easy to recognize these objects. So this is the role of context. This has to do with the role of context in vision. This is something that we don't understand well at all. How do we go from objects to scenes? How do we incorporate context? I think I have another example. This problem is exacerbated if you present images very fast, as we normally do in many of our experiments. Let me see if this works now. I think I'm going to flash an object very quickly. I'm going to, and the question is again, trying to decipher what the object is. You fixate here, and then I tell you this is where the object is going to be. So you fixate in this location. Let's see if this works. That's it. So that was the object, okay? And I would argue that it's probably very hard. If you were able to guess what this is, I applaud you. You're a genius. You're amazing. Most people cannot. It's super hard. Now, if I do the same thing, but I flash the full image, let me see if I can do that again. Okay, so that's the object. That's the full image. So even though I flashed the image very, very briefly, you might have been able to guess what that object is in that case. So this is because your visual system somehow is able to incorporate a lot of information about the scene. So context plays a crucial role in visual object recognition. We have very limited ways to understand how context impacts visual recognition. This is the full image now. And in the interest of time, I'm going to skip this slide. We've been doing a lot of work trying to build initial models of how to incorporate contextual information. So if you're interested, you can scan this QR code. You can go to our website. This is something that we actually, our lab presented last week in CVPR. But there's plenty of more work to do here in terms of understanding context in recognition. So here's another problem that we have in computer vision right now. And that has to do with the fact that many models use contextual information in what I think is the wrong way. So here's one typical computer vision challenge, which has to do with action recognition. This is the so-called UCF 101 action recognition dataset. This is a dataset that consists of 101 different action categories. Those action categories include applying eye makeup, applying lipstick, playing piano, horse races, soccer penalty, long jump, etc., etc., etc. So there are 101 different actions. There are videos, and the goal is to write code such that the computer vision system will be able to guess what the action is. It turns out that one of the problems with this kind of data sets is that there's a lot of confounding factors. For example, if you just take the first frame of every video and you just take the pixel level context, you can already do well above chance. For example, if there's a lot of green in the image, it turns out that soccer is green, basically. So green is associated with horse races, with soccer penalty, maybe with baseball pitch, okay? But you can already say that very few people are applying makeup in a situation where there's a huge amount of green in the image. Basketball dunks are rarely associated with a lot of green. So just by doing very simple tricks, you can get well above chance in action recognition. This does not mean at all that the network knows anything about recognition. It just means that it capitalizes on some of these very simple contextual cues. So we spent a lot of time trying to come up with better controlled data sets in computer vision. So here's a much more challenging action recognition data set that we created. So here we're asking people to recognize whether somebody's drinking, yes or no, reading, yes or no, or sitting, yes or no. So these are controlled by, in terms of a lot of things. You cannot just look at the colors and be able to discriminate them. The images are very similar, yet it's pretty obvious to you that this person is reading and he's not reading here. So this has to do with really understanding in a very deep way what the action is. And most of the computer vision models for action recognition fail miserably when you introduce better controlled data sets and where their models cannot capitalize on these very simple cues. Okay, I'm gonna move quickly because we are running out of time here. Just very quickly, another problem that that I think is very challenging is the one about image captioning. Here's one example of a website where you can try this for yourself. You can upload your favorite picture in there. So here's a picture of the Tower of Pisa. You upload that to this captioning bot. The goal of this bot is to be able to come up with a short description of the image. And what it does is pretty impressive. It says, I think it's a group of people standing in front of the Leaning Tower of Pisa. So that's pretty good. This is way beyond where we were a decade ago. So this is a major accomplishment of computer vision. And yet I would contend that we're still very far from real scene understanding. So if you upload this picture here, the output is essentially the same. I think it's a person standing in front of the Leaning Tower of Pisa. And this is very problematic because I think that there's much more here than a person standing in front of the Leaning Tower of Pisa. So this person is, this picture is meant to be somewhat funny. The Tower of Pisa is meant to be an ice cream. This is meant to be a cone. She's sticking out her tongue and licking the Tower of Pisa as if it were an ice cream cone and so on. This is really, so saying that there's a person standing in the Leaning Tower, in front of the Leaning Tower of Pisa, I think it's correct in a way, but I think this is not what we'd like to understand. And any human can understand that there's much more going on here. We're nowhere even close to be able to write computer code that will capture what's going on in this type of images. So just to summarize, I told you about attractor-based recurrent neural networks that can perform pattern completion. I gave you a glimpse of how top-down signals can be used to guide eye movements. And I also gave you a couple of very quick examples to argue that we have a very long and exciting way to go towards general intelligence. I am convinced that we will solve this problem. And our bet is that we need to understand better neuroscience, that we need to understand better how our brains solve the problem in order to build better computational algorithms. But there's plenty for all of you to do. I think there are many surprises left, and there's going to be exciting new developments in the coming years, coming up with better and better algorithms that are inspired by a deep understanding of neural circuitry to be able to solve visual tasks. So I think that's pretty close to 1:30. So I'm going to stop there and take any questions.